Bismillahirrahmanirrahim. So in this lecture, we will discuss about the predictive methods using protein sequences. And as we know that uh, we can extract a lot of information from protein sequences, and one of them is secondary structure prediction by using the protein sequence of amino acid in the protein. Secondary structure, as we know that the secondary structures are alpha helices, beta sheet or beta strands, and loop regions. Different amino acids tend to prefer being contained in different secondary structure. Okay. Some amino acid like to be in alpha helices, some amino acid like to be in beta sheets, and so on. And this is because of the emphasis of local sequence information. So several computer programs has been made and available for predicting secondary structure of proteins, and those programs basically rely on these methods. Rely on these methods. And some of them, there are many computer programs for secondary structure prediction, and some of them are famous. Of some of the those programs are among them are PhD SEC, PhD SEC, and or Prof SEC for secondary structure prediction. And there is another program called SciPred, PSIPred, which is similar to the PhD program. The PhD SEC or ProfSec programs are based on machine learning approach, and SciPred program is based on neural network prediction method. So these programs are protein secondary structure prediction programs. There is another aspect of protein structure which is called as solvent accessibility. Solvent accessibility. What is solvent accessibility? Is the area of protein surface that is exposed to the solvent. Protein molecule and the surface area which is exposed to the surface. So the accessible region of the proteins interact. Accessible region of the protein is important because these regions interact with other proteins, other molecules, other metal ions on substrates and ligands, so they are important. And these the surface area is measured in square angstroms. In square angstrom. If a surface area is 0, square angstrom, okay, then it would be that surface area would be considered as buried inside the protein, and the surface area, if it is 300 square angstrom, then that area would be considered as exposed residues. There are several programs for solvent accessibility prediction of the protein. Only these programs need protein sequence, and they will provide that which area it would be in the sequence, which area would be solvent exposed and which area would not be solvent exposed. The program names are PhD ACC, Prof ACC, and JPRED. These are the 3 programs. There are other as well. I have mentioned only, for example, 3 programs which have been used extensively to predict the solvent accessibility of the protein. Okay. Next. Okay, there is another aspect of protein which needs our attention, and this aspect is called as transmembrane segment, transmembrane protein. You see, there are membrane proteins are very important in nature. They are involved in they are involved in cellular interaction. They are involved in drug action. There are many membrane proteins which interact with the drugs, which interact with hormones and so on. There are many, so the trans proteins are very important. So it is important. Sorry, ये किसी की please switch off the microphone. Please switch off the microphone. Okay, there are two types of two classes of the membrane protein. One class is called as those one. There are two classes. Those that insert helices into the lipid bilayer, and those that insert alpha beta sheets or strands. So the proteins, membrane proteins, two types of membrane proteins. One, those that insert their alpha helical region in the lipid bilayer of the cell, and the other type of protein which insert their their beta strands in the cell. So the physicochemical properties, physicochemical requirement for a residue to be inserted in the membrane is hydrophobicity. So it means that those amino acids which form, which are buried in the membrane, those amino acids should be hydrophobic in nature, because the lipid bilayer is also hydrophobic in nature. So on this basis, there are several programs, computer programs, which predict transmembrane segment in the protein sequences. Okay, so without any other information, without any other data, only on the basis of amino acid sequence, we can predict the transmembrane region in the of the protein, and there are several programs. Some of them, two of them is written over here. One is top pred program and another is phd htm program. These two programs are available for prediction of transmembrane segment. Okay. Oh, yes. Only from the information of protein sequence, one can also provide some information about the protein function only from the protein sequence. And this is because of the similarity at the sequence level implies similarity of function. If two sequences are similar in nature, if the sequence of two proteins are similar in nature, it means that their function would be similar. So only on the basis of protein sequence, you can say a lot about the protein function. Okay. Also, we can also find out different motifs or different patterns of sequence in the proteins. And these patterns, this is one pattern over here, which can provide us information about the structure and function of a particular protein, of a protein which contains this pattern. And this pattern is saying that the first amino acid would be I. I means isoleucine. Second amino acid would be valine. Third amino acid would be glycine. Fourth amino acid would any amino acid can be there. And the fifth amino acid is glycine and threonine and so on. So this is a pattern. Motif is basically a small structural unit of a protein which is composed of two or more than two secondary structure. So this is called motif in protein structure. There are several methods by which we can find out motifs and patterns in protein sequences, and one of them is famous of them is called as ProCite, PFAM, Interpro, and blocks. These are the that is the computer approaches or tools by which you can find out motifs and patterns in protein sequences. Like this one, you see IV, GXGT, LIV, MF, 2X, and GS. 2X का मतलब यहाँ पर है कि any of the two amino acid. This is one of the this is an example of a pattern present in a particular protein with particular function and particular structure. Okay, and yes. So in this lecture, we have discussed a brief overview. That is, in this lecture, we have discussed that using the protein sequences, protein sequence, we can predict the secondary structure in the protein, of the protein, and these are the programs for doing secondary structure prediction. And we also can predict the solvent accessible region of the protein, whether this region or this amino acid in the sequence would be buried inside the protein or exposed to the solvent or not. And we can also predict transmembrane segment in the protein using the sequence information, and also we can tell a lot about protein function using the protein sequence, and also about different motifs and patterns can be talked about using the protein sequence. So these are the predictive methods using protein sequences.